What is AAV, adeno-associated virus? Have you ever wondered how scientists are using tiny viruses to deliver medicine? One such virus is called adeno-associated virus, or AAV. This small virus naturally infects humans and some primates. It belongs to a group known as Dependoparvovirus, which is part of the Parvoviridae family. AAV particles are about 26 nanometers wide, making them incredibly small. They consist of a protein shell, known as a capsid, that surrounds a single strand of DNA. This DNA is linear and relatively short, measuring around 4.7 to 4.8 kilobases in length. What makes AAV interesting is that it does not cause disease in humans by itself. It was first discovered as a harmless contaminant in laboratory samples of adenovirus, which is how it got its name. In the world of biotechnology and nanotechnology, AAV is best known for its role in drug delivery systems and gene therapy. Scientists have modified AAV to create recombinant AAV vectors. These are simplified versions of the virus that can carry specific genetic instructions into cells. To create these vectors, researchers remove most of the virus's own genes and replace them with a gene or DNA sequence that is of interest, like a therapeutic gene for treating a genetic disorder. The process of making these vectors involves several steps that resemble modular computing or software engineering. Researchers use separate DNA components, often referred to as plasmids, to provide the necessary parts for virus assembly. One plasmid carries the gene to be delivered, another provides the proteins needed to build the virus shell, and a third supplies helper functions from a different virus, usually adenovirus, that AAV needs to replicate. This modular approach allows for flexibility and customization, similar to assembling code from different libraries in software development. AAV vectors have several properties that make them appealing for gene delivery. They can infect both dividing and non-dividing cells. Once inside, the delivered DNA typically remains separate from the host cell's own DNA, which reduces the risk of disrupting important genes. While AAV's small size limits how much genetic material it can carry, it is often sufficient for many therapeutic genes, especially those used in optogenetics. This field involves expressing light-sensitive proteins in cells to control their activity with light. In practical terms, AAV-based systems are being tested and used in clinical trials for diseases affecting the eyes, muscles, nervous system, and liver. Their ability to deliver genes efficiently and with low toxicity has made them a leading choice in developing next-generation therapies. From a technology perspective, the design and production of AAV vectors can be viewed as a form of biological programming. Here, viral components are repurposed to execute specific genetic instructions within living cells. While AAV itself is not a computing technology, the principles behind its use, modular design, programmability, and delivery of functional payloads, parallel concepts in software and hardware engineering. This overlap makes AAV a compelling example of how biological systems can be harnessed and optimized using approaches inspired by computing and technology. As research continues, the role of AAV in medicine and biotechnology is likely to grow, offering new ways to treat diseases and manipulate biological systems with precision.